Bismillah ar-Rahman ar-Rahim. So today we are going to explain the general view of metabolism and how those sources are metabolized by cytosol of the cells and mitochondria. So to begin with, there are two main major sources of those metabolites. First of all, those metabolites, they can come from the diet itself. So this is the outside sources. And we call that well-fed state. So when the cells in the well-fed state, most of energy sources, they come from outside sources, from the diet. And the second one, when we are in a starvation mood, so the source of energy will come from the storage in the body. So we know that there are three major macromolecules that we use in the cells as a source of ATP: amino acids, triglyceride, and glucose. Okay, so once we get them inside the cells, some of them, they need to be metabolized further into the mitochondria. So what happened, the first procedure, for example, for glucose, it need to be activated and converted to glucose 6-phosphate through an enzyme called hexokinase. When we activate glucose to glucose 6-phosphate, this is a committing step. That means it will prevent the diffuse of glucose outside the cells. Then this glucose 6-phosphate, if we have excess of energy and for one reason this metabolism shut down, so that glucose 6-phosphate will enter into glycogen synthesis and we will store it in both liver and muscle. However, if we need ATP and the ADP is high, ATP is low, for example, so the cells, they will need more energy, glucose will be break down further to other metabolites. It is actually a 10 steps. We'll focus in dihydroxyphosphate, okay? Why is that? Because dihydroxyphosphate is one of the precursors of glycolysis. It can also come from glycerol and triglyceride. We know that triglyceride consists of 3 fatty acid chains and 1 glycerol. The glycerol will convert it to dihydroxyphosphate, which again, if we need glucose, can be converted to glucose or can involve in glycolysis. Okay, so after dihydroxyphosphate will convert it to pyruvate. This 10 step of glycolysis, it will occur in systole, okay? And other source of pyruvate is lactate. And we know that lactate can come from two organs in the body. It can actually come from RBC, red blood cells, because they have no mitochondria, so they cannot break pyruvate further into systole, so they will need to convert it to lactic acid. Or during exercising, it can come from muscle, okay? Because when we exercise, there will be shortage in oxygen. So muscles will be in hypoxic status. That's why, because there is no oxygen, it will be the pyruvate will convert it to lactate. And then this lactate will be then shuttled to liver to convert it to pyruvate to produce glucose and ATP. Okay. This cycle converting lactate to pyruvate is called Cori cycle. Okay. Other source of pyruvate is alanine that come from amino acids, and sometimes muscle they talk to liver through a cycle called glucose alanine cycle. So what happened? Muscle will send alanine to liver to be converted to glucose, and then it will be shuttled back to muscle and used as a glucose. Okay. So then this pyruvate for to be further break down and produce more ATP, because in this process it can produce low ATP. But then if you want to produce more ATP, roughly from 34 to 38, we need to move this pyruvate and shuttle it to mitochondria. There are a specific shuttle in mitochondria that can move pyruvate into mitochondria. And into mitochondria, pyruvate through pyruvate carboxylase can convert it to acetyl-CoA. This enzyme only present in mitochondrial matrix. Okay? So if we look into mitochondria, okay, it has actually two membrane. The membrane, the outside outer membrane, it is actually has a pores all around the mitochondria. This will help the movement of ions, but not metabolites, because the ions they are very small in size and they can go inside mitochondria without any shuttling. But for metabolites such as pyruvate, amino acids, triglycerides, they will need a specific carrier protein. It's called mitochondrial carrier protein that they can carry them inside mitochondria. Okay, so here we show the second mitochondrial membrane. However, this carrier protein for pyruvate is unknown. Okay. So once we convert pyruvate in mitochondrial matrix into acetyl-CoA through pyruvate carboxylase enzyme, acetyl-CoA, okay, it is a 2-carbon. Pyruvate is a 3-carbon. So why we need pyruvate carboxylase? Because we need to remove 1 carbon in the form of CO2, okay, in the form of CO2, and we keep the acetyl-CoA, which is 2-carbon. In order to start TCA cycle, we need another source, carbon source, which is oxaloacetate. Oxaloacetate is a 4-carbon, okay? Will join acetyl-CoA to form a complex called citrate. The enzyme will help this process. It's called citrate synthase. So what happened? We will actually join the two. We will condense both acetyl-CoA and oxaloacetate, which is 4-carbon, with acetyl-CoA 2-carbon to form citrate, which is 6-carbon. Okay, and then citrate will convert it to isocitrate, and then isocitrate will convert it to ketoglutarate. Ketoglutarate will convert it to succinyl-CoA. Okay, and succinyl-CoA will be also converted to succinate, which is a 4-carbon. Succinate will convert it to fumarate, okay, through a special enzyme called succinate dehydrogenase. Because what happened, we remove 2 hydrogen from succinate to form fumarate. This is why we call it succinate dehydrogenase. This enzyme is not present freely in mitochondria. It actually attached to complex 2 in electron transport chain. And this enzyme, it need a coenzyme factor to be work, which is FAD. So what happened? When we convert succinate to fumarate, we remove 2 hydrogen atom, and then if FAD will work as a carrier and it will form FADH2. After, fumarate will convert it to malate, and then malate will produce oxaloacetate again. Some of you could ask where oxaloacetate in the beginning come from. Oxaloacetate has different sources. It can come from amino acid deamination. Some of amino acid can produce oxaloacetate, and it can come from other sources of energy, okay, to feed in TCA cycle. Okay, so as we said, so 6-nate dehydrogenase is actually part of ATC, electron transport chain. So as you can see in the slide now, we are showing the complex. There are 5 complexes. First one on the left side is complex 1, which will be responsible for NAD. Second one, which has 6-nate dehydrogenase, is a second complex. Third one, complex number 3, it's called cytochrome C, and then complex number 4. Okay. And the last one is ATP synthase. We'll talk about it later. Okay, so as you can see in the top side, triglyceride will be, glycerol part will convert it to, as we said, dihydroxyphosphate, which is precursors for glycolysis, while fatty acids, as you can see in the top of the slide, will be converted to acyl-CoA. Fatty acid, the first committing step in fatty acid breakdown is activation of acyl-CoA. So how we activated fatty acid? By joining acyl-CoA. However, the acyl-CoA, which is activated form of fatty acid, cannot enter to the mitochondria. It will need a special carrier. This carrier is called carotene carrier protein. How this carrier protein carry acyl-CoA? So what happened? We converted acyl-CoA to acyl. We remove the CoA, and then in this process, acyl fatty acid will form structures called fatty acid acyl carotene. And then inside the mitochondrial matrix, acyl carotene, repeat all the slide. We need to go back. Okay. So acyl carotene inside the matrix will be converted to acyl-CoA again. Okay. Then when we get acyl-CoA, it will enter to beta oxidation, fatty acid beta oxidation. As you can see, TCA, fatty acid oxidation, both occur in the matrix of mitochondria, and they talk to each other. So why does that happen? Because when we break down any fatty acid, either odd chain 1 or even chain fatty acids, will form acetyl-CoA, which is 2 carbon structures, the end product of fatty acid breaking down. Once we get acetyl-CoA, it will enter into TCA cycle. Okay. However, if we have odd chain fatty acids, we cannot produce acetyl-CoA alone. We'll produce a 3 carbon structure called proponyl-CoA. We'll not talk about it in these lectures. Okay. So once we get the acetyl-CoA from fatty acid, from fatty acid oxidation, if there is no enough oxaloacetate, if there is no enough oxaloacetate, which mainly come from glycolysis, what happened? We're left with acetyl-CoA alone. That's why we can't form citrate, because in order to form citrate, we'll need 4 carbon to join it with acetyl-CoA to form the 6 carbon structures citrate. So what happened to acetyl-CoA? It will accumulate it in mitochondria and then will convert it to ketone bodies. This happens during starvation when we don't have enough glucose. Okay. Second thing, okay, when we have too many acetyl CoA coming inside the matrix of mitochondria that either come from glucose and fatty acid, we are in well-fed state, we are eating too much, there are too much glucose and triglyceride reaching to the mitochondrial, to the liver. So what happened at this state? Citrate will accumulate into mitochondrial matrix. Because it accumulate, there will be too much ATP. ATP will block citrate converting to isocitrate. Okay, so isocitrate dehydrogenase enzyme will be blocked. Okay, and then the citrate, because it's accumulated into mitochondrial, will be shuttled outside the mitochondria through a carrier, mitochondrial carrier protein called citrate. Once we shuttle it outside, citrate will be converted to acetyl CoA again and oxaloacetate. Acetyl CoA, okay, in order to start fatty acid synthesis, which is we need 2 block of acetyl CoA, okay, 2 carbon block to start the synthesis. So for this one, we need a coenzyme which is NADPH. Where does NADPH come from? From the process of converting oxaloacetate to malate. So NADPH will feed the process of acetyl CoA converting and forming fatty acid synthesis through an enzyme called fatty acid synthase. So the procedure will occur outside the mitochondria while the breakdown will be inside. So the reason why this is a regulatory process, why we have synthesis in systole and breakdown in mitochondria. So then this process can be organized, okay? Okay, other source of energy, as you can see, we start talk about amino acids. Mainly, amino acid will be shuttled into mitochondria in the form of glutamate, okay, that will be converted to aspartate. And in this process of deamination, transamination, will form the keto acid for aspartate, which is oxaloacetate, and keto acid of glutamate, which is ketoglutarate. And again, oxaloacetate can be converted to malate and it will enter to the mitochondria through a special shuttle called malate aspartate shuttle. Okay, so covering the malate aspartate shuttle is beyond our topics we want to cover in this lecture, so we'll not talk about it, but it's very interesting topic. Other source of amino acids that can go inside mitochondrial matrix is glutamine. So we normally carry ammonia when there's excess amino acids in the body and we want to get rid of ammonia in the liver or kidney, because this is the 2 main cells that can do urea cycle. We will transport amino acids inside the blood using a carrier amino acid, which is glutamine. Why we don't use glutamate? Because it has a charge, negative charge. If you carry ammonia in glutamate amino acids, will cause lowering the pH of blood. That's why we use glutamine, because it has no charge. Okay, inside mitochondria, glutamine can be converted to glutamate by deamination, removing ammonia. Okay, we can form glutamate. This ammonia, okay, it's called the free ammonia, will form the carbamyl phosphate. Carbamyl phosphate is the committing step of urea cycle. Okay, glutamate, if we have excess of glutamate, will be converted to ketoglutarate and fed the TCA cycle if we need ATP. Okay. The other source of uh, so the TCA precursors, mainly they came from amino acids or can come from fatty acids like succinyl-CoA when we talk about odd chain fatty acid oxidation. Okay, so you can see that in TCA cycle, okay, malate as well can come from aspartate. Where does this aspartate come from? It can come from exchange process between glutamate and aspartate, or it can come from urea cycle. Okay, so as you know that in urea cycle we need two ammonia. One come from free ammonia that's inside mitochondria. We can only do this process inside the mitochondria, and the rest of urea cycle will occur outside in systole, and the other source is aspartate. Okay, now we talk about TCA's electron transport chain. So let's come back to our complex. You can see that in the first complex, complex 1, we convert NADH to NAD. When we do this, when we oxidize NADH, actually we are losing hydrogen atom and 2 electron. We are losing 2 electron. This electron will move inside the complex, okay? Will move inside the complex, will be bumped inside electron transport chain. This is why it's called electron transport chain. So the process of bumping this electron will be helped by hydrogen ion pump. Once we get into complex 4, this electron will be carry in oxygen. So oxygen in complex 4 will work as a carrier for those 2 electrons, okay? The other electrons, they can come from FADH2. When lose 2 hydrogen atom, it can give us 2 electrons as well. So for oxygen, we need to form, converted when it carries this electron, we use the hydrogen to form water. That's it for the lectures. I hope you